There is a persistent idea in modern science communication, sometimes called the blue theory. It is not a formal theory in the sense of equations or predictive models, but rather a summary of a strange chemical and physical observation. Blue appears everywhere in the world we see. The sky appears blue. The ocean appears blue. Many birds, insects, and minerals appear blue, and yet, when chemists analyze the molecular structures responsible for natural color, true blue pigments are extremely rare compared to red, yellow, green, or brown. From a chemistry perspective, this is counterintuitive. If blue is visually common, we would expect blue-colored molecules to also be common. But when we analyze natural organic pigments, we repeatedly see the same pattern. Most biological pigments absorb blue light instead of reflecting it. This means blue wavelengths are often removed from reflected light rather than preserved. The color blue, in many cases, is what remains only when other wavelengths are removed, scattered, or redirected. At the molecular level, color is produced when electrons absorb specific photon energies. The energy difference between electron orbitals determines which wavelengths of light are absorbed. Most stable organic molecules have orbital energy gaps that correspond to absorbing higher energy photons first. Blue light sits at the higher energy end of the visible spectrum. That means blue photons are more likely to be absorbed and used to excite electrons into higher energy states. This creates a problem. High-energy excited states are often unstable. They can lead to photochemical reactions, oxidation, or molecular bond breakage. From a biochemical survival standpoint, molecules that strongly interact with blue light are often damaged by it. Over evolutionary time, organisms tend to favor pigments that absorb lower energy light or distribute absorption across a broader range to avoid damage. Because of this, many naturally occurring molecules that could theoretically appear blue instead appear red, purple, or green. To appear blue through pigment chemistry alone, a molecule must absorb most longer wavelength light while avoiding absorption of shorter wavelength blue photons. Achieving that balance while maintaining chemical stability is extremely difficult using organic carbon-based chemistry. This is where physics enters the story. When chemistry cannot easily produce a stable blue pigment, biology often produces blue through physical structure instead. Instead of relying on molecular absorption, organisms build nanoscale structures that manipulate light using interference and scattering. These structures selectively reflect blue wavelengths while cancelling out others. In these cases, blue is not stored inside a molecule. It is generated dynamically by light interacting with structure. If the structure is destroyed, the blue disappears instantly. This is observed in bird feathers, butterfly wings, and fish scales. Crush the material and the blue vanishes, even though the chemical composition remains. This leads to the core idea behind the blue theory. Blue in nature is often not a chemical identity. It is an optical event. Welcome to the Midnight Lesson. If you enjoy deep science topics explained slowly and clearly, please like the video and subscribe to the channel. If you have ideas for future topics, drop them in the comments. Tonight, we are exploring chemistry, physics, and biology together through a single color that challenges how we think about reality. If blue is often structural rather than chemical, that forces us to reconsider what color actually is. In daily life, we speak as if color is a property of objects. We say the sky is blue, the ocean is blue, a feather is blue, but chemistry tells us something more precise. Objects do not contain color. Objects contain molecular structures that interact with electromagnetic radiation in specific ways. The brain then interprets those interactions as color. This means color exists at the intersection of physics, chemistry, and neurobiology. It is not stored in matter the way mass or charge is stored. It is produced through interaction. Blue is the clearest example of this principle because it frequently emerges only when light, structure, and perception align perfectly. Without light, there is no blue. Without structure, there is no blue. Without an observer capable of detecting blue wavelengths, there is no blue. In this lecture, we will approach blue as a boundary condition. It sits at the edge of what organic chemistry can easily produce and what optical physics can easily generate. By studying blue, we gain insight into molecular orbital theory, photochemistry, solid-state optics, atmospheric chemistry, and even neuroscience. Tonight, we begin with the most fundamental question in chemistry and physics. What is color at the level of electrons and photons? Chapter 1. Electronic structure and the origin of color. To understand color from a chemistry standpoint, we must begin with electrons and energy levels. Color is fundamentally about energy transfer between light and matter. More specifically, it is about electrons absorbing photons and moving between quantized energy states. Atoms contain electrons that occupy orbitals. These orbitals represent probability distributions describing where electrons are most likely to be found. When atoms form chemical bonds, atomic orbitals combine to form molecular orbitals. These molecular orbitals are classified as bonding orbitals, antibonding orbitals, and sometimes non-bonding orbitals. The two most important orbitals for color chemistry are the highest occupied molecular orbital, called the HOMO, and the lowest unoccupied molecular orbital, called the LUMO. The energy difference between these two orbitals determines what wavelength of light can be absorbed. When a photon strikes a molecule, it can be absorbed if the photon energy exactly matches the energy gap between HOMO and LUMO. If the photon energy is too low, it passes through or is reflected. If the energy is too high, it may cause ionization or other photochemical reactions rather than simple excitation. Photon energy is inversely proportional to wavelength. This relationship is described by the equation E equals hc divided by lambda, where h is Planck's constant, c is the speed of light, and lambda is wavelength. Shorter wavelengths correspond to higher energy photons. Blue photons therefore carry more energy than red photons. This is critically important. Molecules that interact strongly with blue light must tolerate higher energy electron excitation. Many organic molecules cannot do this repeatedly without degrading. Organic pigments usually rely on conjugated pi electron systems. These systems occur when alternating single and double bonds allow electrons to delocalize across multiple atoms. Delocalization lowers the energy required to excite electrons. As the conjugated system length increases, the HOMO-LUMO energy gap decreases. Short conjugated systems absorb ultraviolet light. Medium-length systems absorb visible blue or green light. Long systems absorb red, or even infrared light. This relationship can be approximated using a particle-in-a-box model, where electrons confined to a conjugated system behave similarly to particles trapped in a one-dimensional potential well. As the box length increases, energy level spacing decreases. This means longer conjugation leads to absorption of lower energy photons. Carotenoids are a classic example. These molecules contain long chains of conjugated double bonds. Their HOMO-LUMO gap corresponds to absorption in the blue region of the spectrum. Because they absorb blue light, they reflect orange and red light, which is why carrots appear orange. Chlorophyll represents a different strategy. It uses a porphyrin ring system coordinated around a magnesium ion. The conjugated ring system absorbs strongly in red and blue wavelengths, reflecting green. These examples highlight a key principle. Most stable biological pigments are tuned to absorb blue light, not reflect it. Blue photons are energetic and can easily drive electronic transitions in organic molecules. Preventing absorption of blue light while absorbing everything else requires extremely precise molecular engineering. Beyond simple absorption, excited electrons often lose energy through vibrational relaxation or internal conversion. In some cases, they return to ground state by emitting light, which produces fluorescence. In other cases, they transfer energy to surrounding molecules, generating heat or reactive oxygen species. Reactive oxygen species can damage biological tissues. Molecules that strongly absorb blue light are therefore often involved in photoprotection rather than coloration. Melanin, for example, absorbs across a wide spectral range and dissipates energy as heat, protecting cells from radiation damage. From a thermodynamic perspective, molecules that frequently enter high-energy excited states experience greater oxidative stress and structural damage. This makes long-term stability difficult. This is one of the main chemical reasons blue pigments are rare. To appear blue, a molecule must avoid absorbing blue photons while still absorbing longer wavelengths. Achieving that balance is chemically difficult using carbon-based organic frameworks. Another factor is solvation and environment. Many pigment molecules exist in aqueous environments inside cells. Water molecules influence orbital energies through hydrogen bonding and dielectric effects. Small environmental changes can shift absorption spectra significantly. Maintaining stable blue absorption bands in fluctuating cellular environments is extremely difficult. This leads to a general chemical trend. Stable natural pigments tend to appear red, yellow, brown, or green. Blue often requires either metal complexes, highly specialized protein environments, or structural optical effects. From a freshman chemistry perspective, blue teaches us something fundamental. Molecular electronic structure does not just determine reactivity, it determines what parts of the electromagnetic spectrum molecules can interact with safely. In most organic chemistry, stability increases when molecules interact with lower energy photons. Blue sits near the threshold where photon energy begins to overlap with bond dissociation energies and photochemical reaction activation energies. This means that while blue light is common in the environment, stable molecules that reflect blue light are chemically uncommon. And this is where the story begins. Not with color as decoration, but with color as a constraint imposed by quantum mechanics, molecular stability, and photochemistry. Chapter 2. Conjugated systems, molecular orbital tuning, and the chemical limits of blue pigments. In the previous chapter, we established that color at the molecular level is governed primarily by electronic transitions between molecular orbitals. We introduced the HOMO-LUMO gap as the critical factor determining what wavelength of light a molecule can absorb. Now we will go deeper into the chemistry that controls this gap, specifically focusing on conjugated systems, resonance stabilization, and why these systems naturally tend to absorb blue light rather than reflect it. Most natural pigments are organic molecules, built primarily from carbon, hydrogen, oxygen, and sometimes nitrogen. Carbon is uniquely suited for pigment chemistry because it can form stable covalent bonds and create extended networks of alternating single and double bonds. These alternating bonds form what chemists call conjugated systems. In a conjugated system, pi electrons are not confined to a single bond. Instead, they are delocalized across multiple atoms. This delocalization lowers the overall energy of the molecule and allows electrons to move more freely across the structure. Delocalization also directly influences the spacing between molecular energy levels. To understand why this matters, it is helpful to imagine a simplified physical model. In physical chemistry, conjugated electron systems are often approximated using the particle-in-a-box model. In this model, electrons behave like particles confined within a region of space. The longer the region, the closer together the allowed energy levels become. In molecular terms, increasing the length of conjugation increases the effective box length. As this happens, the energy difference between the HOMO and LUMO decreases. When the energy gap decreases, the molecule absorbs lower energy photons, which correspond to longer wavelengths of light. Short conjugated systems typically absorb ultraviolet light, which is invisible to humans. As conjugation increases, absorption shifts into the visible spectrum. Moderate conjugation leads to absorption in the blue region. Very long conjugation leads to absorption in green, yellow, or red wavelengths. This explains why many plant pigments appear yellow, orange, or red. Their conjugation length is tuned to absorb higher energy blue photons. The remaining reflected light appears warm-colored. Carotenoids are a classic example of this behavior. Beta-carotene contains a long chain of alternating double bonds. Its absorption spectrum peaks in the blue region of visible light. Because it absorbs blue light efficiently, the reflected light appears orange. From a molecular stability standpoint, this is ideal. The molecule uses blue photon energy for electronic transitions, but is stabilized by its extended conjugated network. Now consider what would be required for a molecule to appear blue instead. A blue-appearing pigment must reflect blue wavelengths while absorbing longer wavelengths, such as red and green. To do this, the HOMO-LUMO gap must be tuned so that absorption occurs at lower energy levels, not higher ones. This requires a molecular orbital configuration that is unusually precise. The molecule must be complex enough to absorb lower energy photons while somehow avoiding absorption of higher energy blue photons. This is difficult because organic molecules generally have absorption spectra that broaden toward higher energies. If a molecule absorbs red light, It often also absorbs some green and blue light unless the absorption band is extremely narrow. Another complication is vibronic coupling. Molecular vibrations can broaden absorption peaks, making selective absorption difficult. Thermal motion in biological systems further broadens spectral behavior. This makes it even harder for organic pigments to reflect a pure blue color through absorption alone. Photochemical stability adds another layer of difficulty. Blue photons carry significantly more energy than red photons. The energy of a photon is given by Planck's equation, where energy equals Planck's constant multiplied by frequency. Because frequency increases as wavelength decreases, blue photons are more energetic than red photons. This increased energy creates risk. When molecules absorb high-energy photons, electrons may be excited into states that can lead to chemical reactions. These reactions can break bonds, rearrange structures, or generate reactive oxygen species. Reactive oxygen species can oxidize biological molecules and cause cellular damage. From an evolutionary perspective, this means pigments that strongly interact with blue light are often selected for protective roles rather than coloration roles. Melanin is a good example. Melanin absorbs broadly across ultraviolet and visible wavelengths. Instead of producing a bright visible color, it dissipates absorbed energy as heat. This protects biological tissues from radiation damage. To produce a blue pigment, evolution would need to create a molecule that resists absorbing high-energy photons while still producing strong visible color. This is chemically inefficient and risky. Now we can examine anthocyanins as a borderline case. Anthocyanins are flavonoid pigments responsible for red, purple, and sometimes blue coloration in plants. Their base structure is a flavillum ion, which contains a conjugated ring system capable of visible light absorption. Anthocyanin color is highly sensitive to environmental conditions. At low pH, anthocyanins appear red. As pH increases, their structure changes and absorption shifts toward purple and sometimes blue. However, this blue is rarely pure and is often unstable. The shift toward blue often requires metal-ion coordination. Metal ions, such as aluminum or magnesium, can bind to anthocyanins and stabilize specific electronic states. This binding can shift absorption spectra toward longer wavelengths, allowing blue reflection. However, this requires tightly controlled cellular conditions. Even with metal coordination, anthocyanin blues are sensitive to temperature, hydration, and oxidative stress. This instability demonstrates how difficult it is for organic chemistry alone to produce stable blue coloration. Another important factor is solvent interaction. Pigment molecules exist inside cells surrounded by water and other polar molecules. Solvent molecules influence molecular orbital energies through electrostatic interactions and hydrogen bonding. These effects can shift absorption wavelengths unpredictably. Maintaining a stable blue pigment in a fluctuating aqueous cellular environment is extremely challenging. Small changes in pH, ionic strength, or hydration can shift absorption bands significantly. Because of these chemical constraints, biology often uses an alternative approach. Instead of trying to build a molecule that reflects blue light, organisms build structures that manipulate light physically. This represents a shift from chemical color generation to physical color generation. Instead of tuning molecular orbitals, organisms tune nanoscale spacing between materials with different refractive indices. This shift dramatically increases stability. Structural color does not depend on fragile, excited electronic states. It depends on geometry and refractive index contrast. These properties are much less sensitive to photochemical damage. From a thermodynamic standpoint, structural color can be energetically cheaper to maintain once the structure is formed. From an evolutionary standpoint, this makes structural blue highly attractive. Another important chemical concept is excited state lifetime. When electrons are excited by photon absorption, they do not remain excited indefinitely. They relax back to lower energy states through several possible pathways. These include fluorescence, phosphorescence, non-radiative decay, and photochemical reaction. Blue photon excitation often leads to short excited state lifetimes and high probability of non-radiative decay. The absorbed energy is converted into molecular vibration, which generates heat. If energy dissipation pathways are inefficient, reactive intermediates may form. This further discourages evolution from relying on blue-absorbing pigments for coloration. Spectroscopy data supports these trends. Many organic molecules show strong absorption in ultraviolet and blue regions due to pi to pi star transitions, achieving selective absorption only in lower energy regions, while avoiding blue absorption requires highly specialized molecular design, rarely achievable through biological synthesis. In summary, conjugated organic chemistry naturally produces pigments that absorb blue light. Producing pigments that reflect blue light requires extremely precise orbital tuning, resistance to photochemical damage, and environmental stability. These requirements are rarely met in biological systems. This is why blue pigments are chemically rare, unstable, or conditional, and this is why biology frequently turns to physics rather than chemistry to create blue coloration. In the next chapter, we will shift from molecular orbital theory into photochemistry and excited state reaction pathways. There we will examine exactly what happens inside molecules when high-energy photons are absorbed and why blue light can be chemically destructive. Chapter 3. Photochemistry, excited states, and why blue photons drive molecular damage. To fully understand why stable blue pigments are rare in biological systems, we must move beyond static molecular orbital diagrams and examine what happens after a photon is absorbed. This is the domain of photochemistry, the branch of chemistry that studies chemical reactions initiated by light. Photochemistry is fundamentally about excited electronic states, energy dissipation pathways, and the competition between stable relaxation and destructive reaction. When a molecule absorbs a photon, an electron is promoted from a lower energy molecular orbital, typically the HOMO, into a higher energy orbital, typically the LUMO or another excited state orbital. This transition happens extremely quickly, usually on the order of femtoseconds. However, once the electron is excited, the molecule enters a state that is thermodynamically and kinetically unstable compared to its ground state configuration. Excited states can be divided into two main categories: singlet excited states and triplet excited states. In a singlet state, the electron spins remain paired. In a triplet state, spins become unpaired and parallel. Triplet states are generally lower in energy than singlet excited states but are longer lived, meaning the molecule remains reactive for a longer period of time. The transition from singlet excited state to triplet excited state is called intersystem crossing. This process is important because triplet state molecules often participate in photochemical reactions, including energy transfer reactions that generate reactive oxygen species. Blue photons are particularly important in this context because of their energy. The energy of visible photons ranges roughly from 1.6 electron volts for red light to about 3.1 electron volts for violet light. Blue light sits in the higher energy portion of this range, typically around 2.7 to 3 electron volts. This energy overlaps with the strength of many chemical bonds found in organic molecules. Carbon-carbon single bonds have bond energies around 3.6 electron volts. Carbon-carbon double bonds are stronger, but still within range of high-energy photon-driven reaction pathways. While a single photon typically does not directly break bonds under normal biological conditions, repeated excitation increases the probability of bond weakening, rearrangement, or radical formation. Once a molecule is excited by a blue photon, several things can happen. The electron can relax back to ground state by emitting light, which produces fluorescence. The electron can relax through non-radiative decay, converting energy into molecular vibration and heat. Or the molecule can undergo photochemical reaction, which changes its chemical structure permanently. In biological pigments, repeated cycles of blue photon absorption increase the likelihood of photobleaching. Photobleaching occurs when a chromophore undergoes irreversible chemical modification after repeated excitation cycles. This is why dyes fade under sunlight. The same process applies to natural pigments. Another major photochemical risk is the formation of reactive oxygen species. When excited state molecules transfer energy to molecular oxygen, oxygen can be converted into singlet oxygen, a highly reactive form. Singlet oxygen can oxidize lipids, proteins, and DNA. In biological tissues, this leads to cellular damage and aging. Because blue photons are energetic enough to efficiently drive excited state formation, pigments that strongly absorb blue light often become photochemically unstable. This creates evolutionary pressure against relying on blue-absorbing molecules for coloration, especially in organisms exposed to sunlight. From a kinetic standpoint, excited state lifetime is critical. The longer a molecule remains in an excited state, the more likely it is to undergo reaction instead of safe relaxation. Blue photon excitation often leads to higher probability of intersystem crossing and longer-lived triplet states. This increases photochemical risk. Molecules can reduce photochemical damage by rapidly dissipating energy through internal conversion. Internal conversion is a process where electronic energy is converted into vibrational energy within the molecule. This energy is then transferred to surrounding molecules as heat. Melanin is highly efficient at this process. Instead of producing bright color, melanin absorbs energy and converts it safely into heat. This again shows the evolutionary bias. Molecules that interact strongly with blue light are often used as protective absorbers rather than visual pigments. Another important photochemical concept is quantum yield. Quantum yield measures how efficiently absorbed photons produce a desired outcome, such as fluorescence or chemical reaction. In pigment stability, low quantum yield for destructive reactions is desirable. However, blue photon absorption often increases quantum yield for unwanted photochemical pathways. Environmental oxygen concentration further increases risk. In aerobic environments, excited molecules are more likely to interact with oxygen and produce reactive oxygen species. Since most life on Earth exists in oxygen-rich environments, pigments must be resistant to oxygen-mediated photochemistry. Blue-absorbing molecules often fail this requirement. Protein binding can sometimes stabilize pigments. In photosynthetic organisms, pigments are often held in specific protein environments that control electronic transitions and prevent harmful reactions. However, these systems are complex and metabolically expensive to maintain. Using such systems solely for coloration is rarely advantageous evolutionarily. Temperature also influences photochemical stability. Higher temperatures increase molecular vibration and collision frequency, increasing the probability of excited state reactions. This means blue pigments must remain stable across environmental temperature ranges, which is chemically difficult. Solvent polarity plays a role as well. Polar solvents, like water, stabilize certain excited states differently than non-polar environments. This can shift absorption peaks and change photochemical reaction pathways. Maintaining a stable blue pigment inside a living cell requires precise solvent-environment control. Another photochemical process to consider is photoisomerization. Some pigments change shape when they absorb light. While this is useful in vision systems like retinal molecules in eyes, it is undesirable in stable pigments. Blue photon absorption increases probability of photoisomerization events in many organic molecules. Over long time scales, repeated exposure to blue light drives cumulative molecular damage. This leads to pigment degradation, loss of color, and formation of breakdown products that may be toxic. In contrast, structural color systems avoid most photochemical risks. Structural color does not rely on repeated excited state electron cycling. Instead, it relies on elastic scattering and interference of light waves. In elastic scattering, photons change direction, but do not transfer energy into electronic transitions. This means structural color systems can reflect blue light without absorbing it, avoiding photochemical damage. This difference is profound. Pigment-based blue would require molecules to interact with high-energy photons repeatedly without degrading. Structural blue allows organisms to display blue color while minimizing chemical interaction with those photons. From an energetic perspective, structural systems shift the burden from chemical stability to mechanical stability. Maintaining nanoscale structure is easier than maintaining photochemically stable blue pigments in many biological contexts. Spectroscopic measurements confirm these trends. Many organic molecules show strong absorption peaks in ultraviolet and blue regions due to pi to pi star transitions. Blue photon absorption is inherently easy for organic molecules. Avoiding blue absorption while maintaining strong visible coloration is the real challenge. From a freshman chemistry standpoint, the key lesson is this. Color is not simply about which wavelengths are reflected. It is about how molecules survive repeated exposure to light. Blue photons carry enough energy to push molecules into unstable excited states. Over evolutionary time, this selects against stable blue absorbing pigments and favors alternative optical strategies. In biological systems, stability often matters more than visual purity. If producing a blue pigment risks cellular damage or requires high metabolic cost to repair, evolution tends to avoid it. This is why when we see blue in nature, it often emerges from physics rather than chemistry. It is safer for organisms to build nanostructures that reflect blue light than to build molecules that must survive constant high-energy photon bombardment. In the next chapter, we will examine one of the few partial exceptions to these rules, plant flavonoid systems such as anthocyanins, and how plants use pH control, metal-ion complexation, and molecular stacking interactions to push pigment coloration toward blue under very specific conditions. Chapter 4. Flavonoids, anthocyanins, metal complexation, and conditional blue pigments in biological chemistry. In the previous chapter, we established that blue light is chemically challenging for organic molecules because it carries high photon energy, increases the probability of excited state reactions, and promotes photochemical degradation. However, nature does produce some pigments that appear blue under certain conditions. These pigments do not contradict the chemical difficulty of blue coloration. Instead, they demonstrate how complex and conditional blue pigment chemistry must be. The most important class of natural pigments capable of producing blue coloration are anthocyanins. Anthocyanins belong to the broader flavonoid family of plant secondary metabolites. These molecules are synthesized through the phenylpropanoid pathway, beginning with the amino acid phenylalanine. Through enzymatic reactions, phenylalanine is converted into cinnamic acid derivatives, which are further modified into flavonoid backbones and eventually anthocyanin chromophores. The core chromophore of anthocyanins is the flavillum ion. The flavillum ion consists of a 3-ring structure containing oxygen heteroatoms and an extended conjugated pi electron system. This conjugated system allows the molecule to absorb visible light. However, unlike many stable pigments, anthocyanins are highly sensitive to environmental conditions. The color of anthocyanins is strongly dependent on pH. At low pH, typically below pH 3, the flavillum ion remains protonated. In this form, anthocyanins typically appear red because they absorb green wavelengths and reflect red wavelengths. As pH increases, the molecule undergoes structural transformations. Deprotonation changes the electron distribution across the conjugated system. This shifts absorption toward longer wavelengths and can move the apparent color toward purple or blue. This shift is an example of a bathochromic shift, also known as a redshift. A bathochromic shift occurs when absorption moves to longer wavelengths, which correspond to lower energy photons. Conversely, a hypsochromic shift occurs when absorption moves towards shorter wavelengths, corresponding to higher energy photons. However, pH alone rarely produces stable blue coloration. Instead, blue anthocyanin colors often require metal-ion coordination. Metal ions such as aluminum, magnesium, or iron can form coordination complexes with anthocyanin molecules. These metal ions interact with oxygen atoms in the anthocyanin structure, stabilizing certain resonance states and shifting absorption spectra. This process is called chelation. Chelation occurs when a metal ion binds to multiple electron-donating atoms in a molecule, forming a ring-like structure. Chelation can stabilize electron distribution across a chromophore, altering molecular orbital energies and shifting visible color. In blue flowers such as hydrangeas, aluminum ion concentration in the soil directly influences flower color. In acidic soils, aluminum ions are more bioavailable and can be transported into plant tissues. When aluminum ions bind to anthocyanins inside plant cells, the pigment complex shifts toward blue coloration. In neutral or alkaline soils, aluminum availability decreases, and flowers appear pink or red instead. This is an example of conditional pigment coloration. The pigment is not intrinsically blue, it becomes blue only under specific chemical conditions. Another factor influencing anthocyanin color is copigmentation. Copigmentation occurs when anthocyanins interact non-covalently with other organic molecules, often other flavonoids. These molecules stack through pp interactions. Stacking changes electron distribution and can stabilize certain excited states. This often produces additional bathochromic shifts, pushing color toward blue. These molecular stacking interactions resemble aromatic ring stacking seen in DNA-base pairing and other biological aromatic systems. The distance between stacked molecules influences orbital overlap and absorption characteristics. Even with pH control, metal-ion coordination, and copigmentation, anthocyanin blues remain unstable compared to other pigment colors. Environmental stress, ultraviolet exposure, temperature fluctuations, and oxidation can all degrade anthocyanin structure. Another complication is hydration state. Anthocyanins can exist in multiple hydration forms depending on water activity. Hydration changes molecular geometry and alters conjugation patterns. This can shift color unpredictably. Cellular compartmentalization partially solves this problem. In plant cells, anthocyanins are stored in vacuoles. Vacuoles allow plants to control pH, ion concentration, and solvent conditions. This controlled microenvironment allows anthocyanins to maintain stable coloration longer than they would in open solution. However, maintaining these conditions requires metabolic energy. From an evolutionary standpoint, producing stable blue pigments through chemistry alone is expensive compared to producing structural color. Another example of protein-assisted pigment stabilization is found in phycobiliproteins in cyanobacteria and algae. Phycocyanin appears blue and is part of the photosynthetic light harvesting system. However, the blue color is not solely due to the pigment molecule. It is strongly influenced by protein binding. The protein environment modifies the chromophore electronic structure and stabilizes excited states. This again demonstrates the pattern. When biology produces blue chemically, it often requires complex support systems such as protein scaffolds or metal coordination. In contrast, structural blue coloration requires no excited electronic states. It relies purely on optical geometry. Once formed, structural coloration is energetically cheap to maintain. It does not degrade through repeated photon absorption. From a thermodynamic perspective, structural color represents a low-energy maintenance solution. Pigment color often requires continuous synthesis and repair due to photodegradation. Structural color requires only maintaining mechanical integrity of nanoscale structures. There is also an evolutionary signaling component. Blue coloration can be advantageous in mate selection and species recognition. If structural color provides brighter, more stable blue signals than pigments, natural selection favors structural solutions. Spectroscopic analysis of anthocyanins shows broad absorption bands rather than narrow peaks. Broad bands reduce color purity. Structural color systems often produce narrow reflection peaks, resulting in more saturated colors. This further favors structural blue for visual signaling. Another important factor is oxidative stability. Anthocyanins are susceptible to oxidation by reactive oxygen species. Structural coloration materials such as keratin or chitin are more resistant to oxidative degradation than conjugated pigment molecules. Even when blue pigments exist, they often exist alongside structural color systems or protective pigments like melanin. This layered approach combines optical selectivity with photochemical protection. From a freshman chemistry perspective, anthocyanins illustrate a key principle. Organic chemistry can approach blue coloration, but only through delicate balancing of electronic structure, environment, and external stabilization mechanisms. These systems are inherently less robust than pigments that absorb high-energy photons rather than reflect them. If we compare this to synthetic chemistry, we see a similar pattern. Many synthetic blue dyes rely on metal complexes or highly engineered aromatic systems. These dyes often require stabilizers to prevent photodegradation. Biological systems face similar constraints but must operate under mild temperatures, aqueous environments, and limited metal availability. In summary, anthocyanins and related pigments demonstrate that blue pigment chemistry is possible, but fragile. It requires precise pH control, metal ion availability, molecular stacking, and cellular compartmentalization. Even then, blue pigment stability is limited. This reinforces the broader chemical principle. Blue lies near the upper energy boundary of visible light, where organic molecules begin to struggle with stability. When biology needs stable, vivid blue coloration, it often turns to physical optical structures rather than purely chemical pigments. In the next chapter, we will move away from organic pigment chemistry and examine transition metal complexes and why synthetic blue pigments often rely on metal-based electronic transitions rather than purely organic molecular orbitals. Chapter 5. Transition metal complexes, ligand field theory, and why true blue pigments usually require inorganic chemistry. Up to this point, we have focused mostly on organic pigment chemistry. We have examined conjugated pi electron systems, HOMO-LUMO transitions, photochemical stability, and the conditional nature of anthocyanin-based blues. Now we shift into an area of chemistry where blue coloration becomes far more chemically accessible. That area is transition metal coordination chemistry. Transition metals occupy the center of the periodic table, primarily in the D block. Unlike main group elements, transition metals possess partially filled d orbitals. These d orbitals are central to color production because they allow multiple low-energy electronic transitions that fall directly within the visible spectrum. To understand why transition metals are so effective at producing blue pigments, we need to introduce crystal field theory and ligand field theory. Crystal field theory describes how the presence of surrounding ligands, which are molecules or ions that bind to a central metal atom, affects the energy of the metal's d orbitals. Ligand field theory expands on this concept by incorporating molecular orbital theory and covalent bonding interactions. In an isolated transition metal ion, the 5 d orbitals have equal energy. This is called degeneracy. When ligands approach and bind to the metal ion, the electric fields generated by ligand electron clouds interact with the d orbitals. This interaction breaks degeneracy and splits the d orbitals into groups of different energy levels. In an octahedral coordination complex, which is one of the most common coordination geometries in chemistry, 6 ligands surround the metal ion. The d orbitals split into 2 sets. The lower energy set is called the t orbitals. The higher energy set is called the Eg orbitals. The energy difference between these sets is called the crystal field splitting energy, often written as delta. If a photon with energy equal to delta strikes the complex, an electron can be excited from a lower energy d orbital to a higher energy d orbital. Because delta often falls within the visible spectrum, these d-d transitions produce color. The exact wavelength absorbed depends on several factors. The identity of the metal ion is important. Different metals have different nuclear charges and electron configurations. The oxidation state of the metal also matters. Higher oxidation states usually increase crystal field splitting. The identity of the ligands is equally critical. Some ligands produce strong crystal fields, others produce weak crystal fields. The spectrochemical series ranks ligands by how strongly they split d orbitals. Strong field ligands produce large energy splitting. Weak field ligands produce smaller splitting. If the splitting corresponds to absorption in the red region, the complex may appear blue because blue light is reflected. This is fundamentally different from organic pigment chemistry. Organic pigments rely on extended conjugation to tune HOMO-LUMO gaps. Transition metal complexes rely on localized electronic transitions within the d orbitals of the metal center. These transitions are often more tunable and more stable under repeated photon exposure. Another important type of transition in metal complexes is charge transfer transitions. In ligand to metal charge transfer, electrons move from ligand orbitals into metal orbitals. In metal to ligand charge transfer, electrons move from metal orbitals into ligand orbitals. Charge transfer transitions are often much more intense than d-d transitions because they involve significant redistribution of electron density. Many intense blue inorganic pigments rely on charge transfer transitions rather than simple d-d transitions. For example, Prussian blue contains ions linked by cyanide bridges. Its intense color arises from electron transfer between ions through the cyanide network. This produces strong absorption in red and green wavelengths, leaving blue reflection. Ultramarine blue, historically derived from lapis lazuli, is another example of inorganic blue pigmentation. Ultramarine contains sulfur radical anions trapped inside an aluminosilicate lattice. These trapped sulfur species absorb specific wavelengths and produce a stable blue color. The crystal lattice protects the chromophore from chemical degradation. Cobalt blue pigments rely on cobalt ions embedded in oxide crystal lattices. The rigid lattice structure stabilizes electronic transitions and prevents photochemical breakdown. These pigments are highly resistant to heat, light, and oxidation. From a chemical stability standpoint, inorganic crystal pigments are extremely robust. The lattice structure distributes photon energy across a solid network. This prevents localized bond damage that would occur in organic molecules. However, biology rarely uses transition metal pigment strategies. There are several reasons for this. First, transition metals are often toxic at high concentrations. Iron, copper, cobalt, and nickel can participate in redox cycling reactions that generate reactive oxygen species. In biological systems, uncontrolled metal redox chemistry can damage cells. Second, building crystalline inorganic lattices inside biological tissues is difficult. Crystal growth requires precise atomic positioning and often high temperature or specific chemical conditions. Biological systems operate at relatively low temperatures and in aqueous environments, which makes forming stable inorganic crystal pigments difficult. Third, maintaining high concentrations of transition metals requires specialized transport proteins, storage systems, and detoxification pathways. This creates metabolic cost and risk. Evolution tends to avoid solutions that require high concentrations of potentially toxic elements. Some organisms do use metal-based coloration in limited ways. Certain mollusks and microorganisms incorporate metal ions into structural materials. Some bacteria produce metal-containing pigments. However, these cases are rare compared to organic pigment or structural color strategies. Another important concept is ligand exchange. In biological systems, metal complexes are constantly exposed to competing ligands, such as water, proteins, and small organic molecules. This can destabilize metal pigment complexes. Synthetic pigments avoid this problem by locking metals inside rigid crystal lattices. Thermodynamics also plays a role. Many transition metal pigments are thermodynamically stable only under conditions not commonly found inside living cells. Biological systems favor dynamic chemical equilibrium rather than rigid crystal formation. If we compare this to synthetic dye chemistry, we see that humans often use transition metal complexes to produce stable blue dyes and pigments. However, these dyes often require stabilizers and controlled environments. Even synthetic blue dyes can fade under prolonged ultraviolet exposure. Another advantage of structural blue coloration over metal pigments is weight. Heavy metal pigments increase tissue density. Structural coloration uses lightweight biological polymers, such as keratin and chitin combined with air pockets. This is advantageous for flying organisms such as birds and insects. From an optical standpoint, transition metal pigments can produce extremely pure and saturated blue colors. However, structural color can produce equally vivid blue while avoiding metal toxicity and photochemical instability. From a freshman chemistry perspective, transition metal complexes illustrate a key principle. The periodic table strongly influences what colors are chemically accessible. Main group organic chemistry struggles to produce stable blue pigments. Transition metal chemistry can produce blue pigments more easily because d orbital splitting naturally falls within visible energy ranges. However, biological constraints such as toxicity, solubility, metabolic cost, and environmental variability make transition metal pigment strategies less practical for most organisms. This leads to a broader chemical insight. Blue is not chemically impossible. It is chemically expensive and risky in organic biological systems. When inorganic crystal chemistry is available, blue pigments become much easier to produce and maintain. But living organisms rarely operate under conditions that favor inorganic crystal pigment formation. This explains why synthetic paints and minerals often contain brilliant blue pigments, while biological systems often rely on structural color for blue appearance. In summary, transition metal chemistry provides a pathway to stable blue coloration through d orbital transitions and charge transfer processes. Inorganic crystal lattices stabilize these electronic transitions and protect chromophores from degradation. However, biological constraints limit the use of metal-based pigments in living systems. Because of these constraints, evolution frequently chooses structural optical solutions instead of inorganic pigment chemistry. Structural color avoids photochemical degradation, avoids heavy metal toxicity, and can be produced through self-assembly of biological polymers. In the next chapter, we will move fully into physical chemistry and optics, examining how refractive index contrast, thin film interference, and nanoscale periodic structures create structural blue coloration without requiring pigment absorption at all. Chapter 6. Refractive index, thin film interference, and the physical chemistry of structural blue. Up to this point, we have examined why organic chemistry struggles to produce stable blue pigments and why inorganic transition metal chemistry can produce blue but is rarely practical for biological systems. Now we move into the physical chemistry that dominates natural blue coloration. Structural color is fundamentally an optical phenomenon governed by electromagnetic wave behavior, refractive index contrast, and nanoscale spatial periodicity. Structural color occurs when light interacts with microstructures or nanostructures that are similar in size to visible wavelengths. Instead of absorbing photons and promoting electrons into excited states, structural color systems manipulate how light waves propagate, reflect, scatter, and interfere. This allows organisms to produce vivid blue coloration while avoiding photochemical instability. The first concept we must define is refractive index. The refractive index of a material describes how much light slows down when traveling through that material compared to vacuum. It is defined as the speed of light in vacuum, divided by the speed of light in the material. When light transitions between materials with different refractive indices, part of the light reflects and part transmits. In biological systems, refractive index contrasts often occur between protein matrices, air pockets, lipids, and water. The magnitude of this contrast determines how strongly light is reflected. Greater refractive index difference leads to stronger reflection. However, reflection alone does not produce color. Color requires wavelength selective reflection. This is where periodic structure becomes critical. When layers of materials with different refractive indices are arranged at regular intervals, light reflecting from each interface can interfere constructively or destructively. Constructive interference occurs when reflected waves reinforce each other. Destructive interference occurs when waves cancel each other out. The condition for constructive interference depends on layer thickness, refractive index, and wavelength of light. For thin film interference, the basic relationship is that constructive interference occurs when twice the optical thickness of a layer equals an integer multiple of the wavelength. Optical thickness is the physical thickness multiplied by refractive index. This means biological systems can tune reflected wavelength by adjusting layer spacing or material density. Blue light has wavelengths roughly between 450 and 495 nanometers. To reflect blue efficiently, biological structures typically require layer spacing around 1/4 to 1/2 of that wavelength when accounting for refractive index. This places required structural spacing firmly within nanoscale biological manufacturing capabilities. Protein-based biological materials such as keratin and chitin naturally self-assemble into structures within this size range. This is one reason structural blue is common in nature. Biology can easily produce structures optimized for blue reflection, but producing structures optimized for red reflection requires larger spacing that may be mechanically unstable or developmentally difficult. Thin film interference can be observed in soap bubbles and oil films. In biological systems, similar principles occur in bird feather, barbules, and insect wing scales. Multiple layers reflect light repeatedly, producing wavelength selective reflection. Another important optical concept is coherent scattering. In coherent scattering, scattered waves maintain fixed phase relationships. This allows interference patterns to emerge. In structural color systems, coherent scattering allows specific wavelengths to be strongly reflected while others cancel out. In contrast, incoherent scattering occurs when phase relationships are random. This produces white or diffuse color, rather than vivid spectral color. Biological structural color systems often balance order and disorder to produce consistent coloration across viewing angles. Perfectly ordered photonic crystal structures produce iridescent colors that change dramatically with viewing angle. Many biological systems use quasi-ordered structures instead. Quasi-order maintains wavelength selectivity while reducing angle dependence. This produces more stable coloration from multiple viewing directions. Photonic crystals represent an advanced structural color system. In photonic crystals, refractive index varies periodically in 3 dimensions. This creates photonic band gaps, where certain wavelengths cannot propagate through the structure. Instead, those wavelengths are reflected strongly. Photonic band gap behavior is analogous to electronic band gaps in semiconductors. Just as electrons cannot occupy forbidden energy ranges in semiconductor crystals, photons cannot propagate through forbidden wavelength ranges in photonic crystals. Biological photonic crystals are often formed through self-assembly rather than direct genetic encoding of nanostructure shape. During development, diffusion, phase separation, and protein polymerization create periodic nanoscale structures naturally. This self-assembly is driven by thermodynamics. Structures that minimize free energy tend to form spontaneously under correct conditions. Evolution then refines these processes to produce reliable coloration. Another advantage of structural color is durability. Because structural color relies on physical arrangement, rather than molecular excited states, it is far less vulnerable to photobleaching. Photons are reflected elastically rather than absorbed. This prevents photochemical degradation. Structural color also provides extremely high color purity. Because interference selectively reflects narrow wavelength bands, structural colors can appear more saturated than pigment colors. This is especially noticeable in blue structural colors in birds and insects. Melanin often plays a supporting role in structural color systems. Melanin absorbs stray scattered light. This increases contrast and prevents washed-out coloration. Without melanin, structural colors can appear pale or white. Temperature stability is another advantage. While thermal expansion can slightly alter structural spacing, biological materials are often flexible enough to maintain functional periodicity across temperature ranges. Hydration can influence refractive index contrast. Some structural color systems change color when hydrated or dehydrated. This demonstrates that structural color depends strongly on material optical properties rather than chemical composition. From an energy standpoint, structural color can be metabolically efficient. Once the structure is built, it requires little maintenance. Pigments must often be continuously synthesized and repaired. Structural blue coloration is common in birds such as blue jays, where keratin and air nanostructures produce wavelength selective scattering. In butterflies such as morpho species, wing scales contain multilayer ridges that produce intense blue reflection through interference. Fish use similar principles with guanine crystal layers inside specialized cells called iridophores. These crystals create reflective multilayer stacks tuned to specific wavelength. From a freshman chemistry perspective, structural blue represents an intersection of chemistry, physics, and materials science. Chemistry controls what materials are available. Physics controls how light interacts with those materials. Biology controls how structures are built and maintained. Structural blue coloration shows that sometimes the easiest way to solve a chemical problem is to avoid chemistry entirely and use physics instead. Instead of building molecules that must survive high-energy photon absorption, organisms build structures that redirect photons without absorbing them. This represents a shift from electronic energy transitions to wave interference as the dominant mechanism of color production. In the next chapter, we will expand structural color concepts into fully three-dimensional photonic crystal systems and examine how biological organisms produce complex optical materials that rival engineered optical devices. Chapter 7. Three-dimensional photonic crystals, quasi-ordered nanostructures, and biological optical materials at the edge of materials science. In the previous chapter, we examined thin film interference, refractive index contrast, and multi-layer reflectors as the foundation of structural color. These systems represent relatively simple optical architectures, typically involving stacked layers or repeating two-dimensional structures. In this chapter, we move into a more complex and more powerful optical regime. Here, we examine three-dimensional photonic crystals, quasi-ordered photonic structures, and biological nanomaterials that function as advanced optical devices. Three-dimensional photonic crystals are materials in which refractive index varies periodically in all three spatial dimensions. This periodic variation creates what is called a photonic band structure. Just as electrons in a crystalline solid experience allowed and forbidden energy ranges due to periodic atomic potentials, photons moving through a photonic crystal experience allowed and forbidden wavelength ranges due to periodic dielectric structure. To understand this concept, it is helpful to recall electronic band theory. In a crystalline semiconductor, atoms are arranged in repeating patterns. Electron wave functions overlap between neighboring atoms. This creates continuous energy bands separated by band gaps. Electrons cannot exist within band gaps. This is the foundation of semiconductor behavior. Photonic crystals operate on an analogous principle. Instead of atomic potentials influencing electron waves, refractive index periodicity influences electromagnetic waves. When periodic spacing matches roughly half the wavelength of light, strong Bragg scattering occurs. If scattering is strong enough across multiple directions, certain wavelengths cannot propagate through the material. These wavelengths form photonic band gaps. If blue wavelengths fall inside a photonic band gap, blue light will be strongly reflected. Other wavelengths will pass through or be absorbed. This produces extremely pure structural color with high intensity and narrow spectral bandwidth. Unlike thin film interference, which usually reflects a narrow range of wavelengths only along specific angles, three-dimensional photonic crystals can reflect light more isotropically. This means color can remain consistent across multiple viewing directions. In biological systems, perfect three-dimensional photonic crystals are rare, but partial or quasi-photonic crystals are common. Quasi-ordered systems contain short-range periodicity without perfect long-range crystal order. These systems still produce photonic band gap-like behavior, but with broader spectral reflection and less angular sensitivity. This quasi-ordering is extremely important for biological coloration. Perfect crystals produce highly iridescent color that changes dramatically with viewing angle. While useful for some signaling behaviors, excessive iridescence can reduce visual reliability. Many organisms need consistent coloration for camouflage or species recognition. Quasi-ordered photonic structures strike a balance. They produce strong wavelength selective reflection while maintaining consistent color appearance from multiple viewing directions. From a materials science perspective, these structures are extraordinary. Biological systems assemble nanoscale photonic architectures using self-organizing biochemical processes. No external fabrication tools are required. Instead, cells control polymerization, phase separation, and molecular diffusion to generate optical materials. One mechanism underlying photonic structure formation is spinodal decomposition. In spinodal decomposition, two materials separate spontaneously into interpenetrating phases when thermodynamic conditions change. In biological tissues, protein and water phases can separate into nanoscale domains. If domain spacing falls within visible wavelength scales, structural color can emerge. Another mechanism is block copolymer self-assembly. Some biological polymers behave similarly to synthetic block copolymers, where chemically distinct segments separate into repeating nanostructures such as lamellae, cylinders, or gyroids. Gyroid structures are particularly interesting because they can produce isotropic photonic band gaps. The gyroid structure is a continuous, triply periodic minimal surface. It divides space into two interwoven networks. Synthetic materials scientists actively study gyroid photonic crystals because of their optical efficiency. Remarkably, some biological systems appear to produce gyroid-like photonic nanostructures naturally. The development of these structures is governed by thermodynamics and kinetics. Cells produce structural proteins or polysaccharides at controlled rates. Diffusion, polymerization speed, and local chemical gradients determine final structure. Once formed, these structures are often locked into place by cross-linking or dehydration. Another important structural color mechanism involves colloidal photonic crystals. In these systems, spherical nanoparticles self-assemble into repeating lattice arrangements. If particle size is comparable to visible wavelengths, Bragg diffraction can produce structural color. In biological tissues, protein nanospheres or air voids can behave like colloidal particles. If these particles self-assemble into face-centered cubic or hexagonal close-packed lattices, structural color can emerge. Defects in these lattices broaden reflection spectra and reduce iridescence. Biological systems often incorporate controlled defect density to tune optical performance. Mechanical properties are also important. Biological photonic structures must survive bending, stretching, and impact. Materials such as chitin and keratin provide both optical function and mechanical strength. This dual function is rare in synthetic photonic materials, which are often fragile. Another remarkable aspect of biological photonic crystals is hierarchical structure. Many organisms use multiple optical layers simultaneously. For example, one layer may produce broadband reflection, while another produces narrowband interference. This layered optical design increases color intensity and complexity. Thermal stability is enhanced by flexible polymer networks. While inorganic photonic crystals often require rigid crystal lattices, biological photonic structures can deform without losing optical function. This resilience allows organisms to maintain coloration across temperature and humidity changes. Another optical phenomenon present in biological photonic materials is multiple scattering. In highly ordered photonic crystals, scattering is predictable and directional. In quasi-ordered structures, multiple scattering can increase brightness while preserving wavelength selectivity. This produces bright but non-iridescent color. Melanin often appears beneath photonic structures. Melanin acts as an optical absorber. It prevents light from passing through the structure and reflecting randomly from deeper tissue layers. This increases contrast and enhances color saturation. From an evolutionary standpoint, photonic nanostructures provide several advantages. They produce bright color without requiring toxic metal accumulation. They resist photochemical degradation. They can be grown using existing biochemical pathways. They provide mechanical structural support. Another advantage is energetic efficiency. Once formed, photonic structures require no metabolic maintenance. Pigments must often be resynthesized as they degrade. Structural colors remain stable and less physically damaged. From a physical chemistry standpoint, these systems represent controlled manipulation of electromagnetic boundary conditions. Maxwell's equations govern how electromagnetic waves propagate through dielectric materials. When refractive index changes periodically, boundary conditions force certain wavelengths to reflect or localize. Localized photonic modes can enhance reflection intensity dramatically. In some biological systems, reflectivity approaches levels seen in engineered mirrors for specific wavelengths. The optical quality of biological photonic materials rivals synthetic materials produced in laboratories. This is remarkable, given that biological systems assemble these materials under ambient temperature and aqueous conditions. Another fascinating area of research is dynamic photonic structures. Some organisms can change structural spacing through hydration, muscular contraction, or pressure changes. This allows dynamic color shifting without changing chemical composition. These systems demonstrate that biological coloration is often a problem in materials engineering rather than pure chemistry. From a freshman chemistry and early materials science perspective, this chapter highlights an important transition. Color is not always determined by molecular identity. It can be determined by nanoscale spatial organization. The same material can appear colorless or vividly colored depending on structure alone. Structural blue coloration is particularly common because required periodic spacing falls within biologically achievable nanometer ranges. Producing structural red requires larger periodic spacing, which is harder to maintain mechanically and developmentally. This creates a natural bias toward blue structural coloration across many evolutionary lineages. In summary, three-dimensional photonic crystals and quasi-ordered nanostructures represent one of the most advanced optical technologies found in nature. These systems produce blue coloration, not by storing color chemically, but by controlling how light propagates through matter. This reinforces the central theme of this lecture. Blue is often not a property of molecules. It is a property of structure interacting with light. In the next chapter, we will shift from biological nanostructures to atmospheric chemistry and molecular scattering physics, examining how gas molecules produce blue sky coloration through wavelength-dependent scattering. Chapter 8. Molecular scattering, atmospheric chemistry, and why the sky produces blue without pigment or structure. Up to this point, we have examined blue through molecular electronic structure, photochemistry, pigment instability, and solid-state optical nanostructures. Now we move into a completely different regime of physical chemistry. In this chapter, blue emerges not from molecules absorbing light, and not from solid structures reflecting light, but from gases scattering light. The blue of the sky represents one of the purest examples of blue existing as an interaction rather than a material property. The atmosphere contains mostly nitrogen gas and oxygen gas, along with small amounts of argon, carbon dioxide, water vapor, and trace gases. None of these gases are blue. None of them contain blue pigments. None of them form photonic crystal structures, and yet collectively they produce a blue sky under sunlight. To understand this phenomenon, we must examine electromagnetic scattering at the molecular scale. When light travels through a medium containing particles much smaller than the wavelength of light, the dominant scattering mechanism is Rayleigh scattering. Rayleigh scattering describes how electromagnetic radiation interacts with particles that are significantly smaller than the wavelength of the incoming radiation. In Rayleigh scattering, the electric field of the incoming light wave induces oscillations in the electron cloud of the molecule. The oscillating electron cloud then re-emits electromagnetic radiation in multiple directions. This re-emitted light has the same frequency as the incoming light, meaning Rayleigh scattering is elastic scattering. The photon energy does not change, only direction changes. The intensity of Rayleigh scattering depends strongly on wavelength. Specifically, scattering intensity is proportional to 1 over wavelength to the 4th power. This means that shorter wavelengths scatter much more strongly than longer wavelengths. Blue light scatters several times more strongly than red light. Violet light scatters even more strongly than blue light. This wavelength dependence is critical. When sunlight enters the atmosphere, shorter wavelengths are scattered in all directions by gas molecules. When you look at the sky away from the sun, you see scattered light rather than direct sunlight. Because blue wavelengths scatter more strongly, the scattered light reaching your eyes appears blue. However, an interesting question arises. If violet scatters more strongly than blue, why is the sky not violet? The answer involves two factors. First, the sun produces less violet light compared to blue light. Second, the human eye is less sensitive to violet wavelengths. Additionally, some violet light is absorbed by ozone in the upper atmosphere. The combined result is that the scattered light appears blue to human observers. From a molecular physics perspective, Rayleigh scattering depends on molecular polarizability. Polarizability describes how easily the electron cloud of a molecule can be distorted by an electric field. Nitrogen and oxygen molecules have electron clouds that respond to incoming electromagnetic waves. The oscillating dipoles formed in these molecules emit radiation efficiently. Because atmospheric molecules are extremely small compared to visible wavelengths, scattering occurs uniformly across many directions. This creates diffuse blue illumination across the sky dome. Another important factor is optical path length. When the sun is high in the sky, sunlight passes through a relatively short atmospheric path. Blue light is scattered but still dominates scattered light reaching the observer. When the sun is near the horizon, sunlight travels through a much longer atmospheric path. During this longer path, much more blue light is scattered out of the direct sunlight beam before reaching the observer. The remaining transmitted light becomes enriched in longer wavelengths, producing red and orange sunsets. This demonstrates that blue sky color is not constant. It depends on geometry, atmospheric composition, and optical path length. Atmospheric scattering can also be modified by larger particles. When particles become comparable in size to visible wavelengths, ME scattering becomes important. ME scattering is less wavelength dependent. This is why clouds appear white. Cloud droplets scatter all visible wavelengths relatively equally. Air pollution also influences scattering behavior. Aerosols can shift sky color by increasing scattering of longer wavelengths. Volcanic eruptions can inject particles into the atmosphere that produce unusually red or purple sunsets. From a thermodynamic perspective, Rayleigh scattering is efficient because it does not require photon absorption. Photons are redirected rather than converted into molecular excitation. This means atmospheric molecules do not experience photochemical stress from scattering visible light. This is another example of blue appearing through physical interaction rather than chemical storage. The atmosphere does not need to produce blue molecules. It only needs to scatter blue wavelengths more efficiently than others. Radiative transfer theory describes how light propagates through scattering media. The radiative transfer equation accounts for absorption, emission, and scattering simultaneously. In the atmosphere, scattering dominates visible wavelength behavior under clear sky conditions. Atmospheric density gradients also influence scattering. As altitude increases, air density decreases. This reduces scattering intensity. This is why the sky appears darker blue at higher altitudes. In near space, where the atmosphere is extremely thin, the sky appears nearly black. Another important atmospheric optical phenomenon is polarization. scattered light becomes partially polarized. Polarization patterns across the sky are used by some animals for navigation. This again demonstrates that scattered blue light carries directional information about the sun's position. From a chemical perspective, atmospheric gases remain chemically stable despite constant exposure to sunlight. Nitrogen and oxygen molecules require ultraviolet photons with much higher energy to break molecular bonds. Visible blue photons do not carry enough energy to dissociate these molecules directly. This allows the atmosphere to scatter blue light continuously without degrading chemically. This is fundamentally different from organic pigment molecules, which can degrade after repeated photon absorption cycles. Another interesting example of scattering-driven blue coloration occurs in smoke and haze. Very small particles can scatter blue wavelengths strongly, producing bluish haze under certain conditions. This again demonstrates that blue often appears when shorter wavelengths are preferentially redirected. Ocean color shares some similarity with atmospheric scattering. Water molecules absorb longer wavelengths more strongly than shorter wavelengths. Blue light penetrates deeper and is scattered back toward the surface. While some ocean blue comes from sky reflection, much of it comes from selective absorption combined with scattering. From a freshman chemistry perspective, the sky provides an example of color emerging from collective molecular behavior rather than individual molecular chromophores. The sky is not blue because nitrogen is blue. It is blue because billions of trillions of nitrogen and oxygen molecules scatter shorter wavelengths more efficiently than longer ones. The sky therefore reinforces the central theme of this entire lecture. Blue often exists not as a chemical property of matter, but as a physical property of light interacting with matter. In atmospheric chemistry, blue is an emergent optical phenomenon produced by statistical scattering events across enormous numbers of molecules. No individual molecule is blue. The bulk system appears blue because of wavelength-dependent scattering probability. This idea challenges everyday intuition. We tend to think of color as something objects possess. Atmospheric blue shows that color can exist purely as the probabilistic optical outcome of light interacting with matter at scale. In the next chapter, we will move from gases back to liquids and examine the optical chemistry of water. We will explore vibrational absorption, hydrogen bonding networks, and why large bodies of water appear blue even when pure water appears colorless in small volumes. Chapter 9. Water, hydrogen bonding networks, vibrational overtones, and the optical chemistry of blue oceans. In the previous chapter, we examined how gases scatter blue light through Rayleigh scattering, producing the color of the sky without pigments or structural nanomaterials. In this chapter, we move into liquid phase physical chemistry and molecular spectroscopy to understand how water contributes to blue coloration in oceans, lakes, and large bodies of liquid. Water appears colorless in small quantities, yet large volumes of water often appear blue. This transition from transparent to blue represents a combination of molecular absorption physics, hydrogen bonding network behavior, and light scattering processes. At first glance, pure water appears colorless because it transmits most visible wavelengths efficiently. However, water is not perfectly transparent. Water molecules absorb electromagnetic radiation through vibrational excitation modes. These vibrational modes arise from stretching and bending motions of the OH bonds within the molecule. Water molecules are not isolated in liquid water. They form dynamic hydrogen bonding networks. Hydrogen bonding occurs because oxygen atoms carry partial negative charge, while hydrogen atoms carry partial positive charge. This leads to temporary electrostatic attractions between neighboring molecules. These networks constantly form and break on picosecond timescales. The hydrogen bonding network modifies vibrational energy levels. Vibrational absorption bands shift and broaden due to intermolecular coupling. In isolated water vapor molecules, vibrational absorption occurs primarily in infrared wavelengths. In liquid water, hydrogen bonding causes vibrational overtone absorption to extend weakly into the red edge of the visible spectrum. This is critical. Liquid water absorbs slightly more red light than blue light. The absorption is weak, but over long distances, even weak absorption becomes significant. This behavior can be understood through the Beer-Lambert law. The Beer-Lambert law states that absorbance increases with path length and concentration. Even if water absorbs only a small fraction of red light per centimeter, over many meters or kilometers, red light is significantly reduced. Blue wavelengths are absorbed less strongly. Therefore, after long path lengths through water, more blue light remains. This selective removal of red wavelengths shifts transmitted and scattered light toward blue. This phenomenon is not structural color and not Rayleigh scattering alone. It is selective molecular absorption combined with scattering processes. Blue color emerges because other wavelengths are removed preferentially. The molecular origin of red absorption in water lies in vibrational overtone transitions. The fundamental vibrational modes of water occur in infrared wavelengths. However, overtone transitions, which involve excitation to higher vibrational quantum states, can extend into visible wavelengths. These overtone transitions are weak individually, but collectively they produce measurable absorption. Temperature influences these vibrational absorption patterns. Increased temperature disrupts hydrogen bonding networks. This shifts vibrational energy distributions and can slightly modify absorption behavior. However, temperature effects are relatively small compared to path length effects. Impurities and dissolved substances significantly influence water color. Dissolved organic matter can absorb blue light, shifting water toward green or brown coloration. Phytoplankton contain chlorophyll, which absorbs red and blue light, often making water appear green. In extremely pure deep water, blue coloration becomes more pronounced. The absence of competing absorbers allows selective red absorption to dominate optical behavior. Another important optical factor is scattering by water molecules and suspended particles. Pure water molecules scatter light weakly, but suspended particles increase scattering intensity. If particle sizes are very small, Rayleigh-like scattering enhances blue reflection. If particle sizes are larger, MIE scattering produces white or milky coloration. Ocean blue therefore results from multiple combined processes. Selective red absorption removes longer wavelengths. Scattering redistributes shorter wavelengths. Reflection at the air-water interface adds additional sky color contribution. However, deep ocean blue is not simply sky reflection. Subsurface scattering and absorption dominate. Optical oceanography measures these processes using absorption coefficients and scattering coefficients. The attenuation coefficient describes how quickly light intensity decreases with depth. Blue light typically penetrates deeper into water than red light. This is why underwater environments appear blue. Another molecular factor influencing water optical properties is density fluctuations. Thermal motion creates microscopic variations in density. These variations can scatter light weakly, contributing to overall optical behavior. Hydrogen bonding also influences refractive index. The refractive index of water depends on temperature, salinity, and pressure. Changes in refractive index affect how light bends and reflects inside water bodies. Salinity increases refractive index slightly. Dissolved salts alter hydrogen bonding networks and electronic polarization behavior. However, salinity effects on color are minor compared to absorption effects. Pressure increases density and refractive index in deep ocean water. However, pressure-induced optical effects are usually small relative to absorption and scattering contributions. Another important factor is surface wave dynamics. Waves change reflection angles and scattering geometry. This can enhance blue appearance under certain lighting conditions. Surface roughness increases diffuse reflection. Biological particles also influence ocean color. Phytoplankton populations can dramatically shift water color. Chlorophyll absorption reduces blue and red wavelengths, increasing green reflectance. This is why productive coastal waters often appear green rather than blue. In extremely clear tropical waters with low nutrient levels, low phytoplankton density allows blue coloration to dominate. These waters appear intensely blue because minimal pigment absorption interferes with pure water optical properties. The interaction between sunlight angle and water depth also influences perceived color. At shallow angles, surface reflection dominates. At direct overhead sunlight, subsurface scattering dominates. From a molecular spectroscopy standpoint, water absorption spectra are well characterized. Absorption increases gradually from blue to red wavelengths. This gradual increase is sufficient to shift color perception over large distances. Another interesting effect occurs in glacial ice. Glacial ice often appears blue because compression removes air bubbles and increases optical path length. This enhances selective red absorption, leaving blue scattered light. Ice optical behavior further demonstrates that water molecules themselves produce subtle spectral filtering effects that become visible only over large distances. From a freshman chemistry perspective, water demonstrates how weak molecular absorption can create strong macroscopic color effects when path length becomes large. This reinforces the principle that color is not always about strong pigment absorption. Sometimes it is about small spectral biases integrated over large distances. Water's blue appearance is therefore an emergent optical property arising from vibrational spectroscopy, hydrogen bonding network physics, scattering processes, and environmental purity. This again reinforces the central theme of this lecture. Blue often appears not because something is chemically blue, but because other wavelengths are preferentially removed or redirected. In the final chapter, we will integrate everything we have learned. We will examine blue as an emergent constraint produced by quantum chemistry, photochemistry, thermodynamics, and optical physics working together across biological and environmental systems. Chapter 10. Blue as an emergent constraint across quantum chemistry, thermodynamics, optical physics, and biological evolution. Throughout this lecture, we have approached blue not as a simple color category, but as a recurring boundary condition imposed by physics and chemistry. In this final chapter, we integrate molecular orbital theory, photochemistry, inorganic coordination chemistry, structural optics, atmospheric scattering, and liquid phase vibrational spectroscopy into a unified explanation. The central conclusion is not that blue does not exist. The more precise conclusion is that blue rarely exists as a stable, intrinsic chemical property of matter. Instead, blue most often emerges from interactions between light, structure, and observation. We begin at the smallest scale, quantum chemistry. At the atomic and molecular level, color originates from electronic transitions. When photons interact with molecules, electrons absorb energy and transition between quantized orbitals. The energy difference between orbitals determines which wavelengths are absorbed. For organic molecules, the dominant color-producing mechanism is pi-electron conjugation. Increasing conjugation lowers the HOMO-LUMO energy gap. Lower energy gaps correspond to absorption of longer wavelength light. This naturally leads to organic pigments absorbing blue light rather than reflecting it. In other words, organic chemistry tends to remove blue wavelengths from reflected light rather than produce them. This is not accidental. It is a direct consequence of quantum mechanical constraints on electron energy levels. Organic molecules are built primarily from carbon-based bonding frameworks. These frameworks produce predictable ranges of electronic transition energies. These ranges often overlap strongly with ultraviolet and blue photon energies. Blue photons carry relatively high energy within the visible spectrum. When molecules absorb blue photons, electrons may enter excited states that promote photochemical reactions. These reactions can produce free radicals, generate reactive oxygen species, or break molecular bonds. Over time, repeated blue photon exposure degrades many organic chromophores. From a thermodynamic and kinetic perspective, this creates strong evolutionary pressure. Pigments that absorb blue photons are useful for photoprotection, but pigments that must survive reflecting blue light repeatedly are chemically difficult to maintain. This is why most stable biological pigments appear red, yellow, green, or brown rather than blue. When biology does produce blue pigment-like behavior, it often requires conditional stabilization mechanisms. Anthocyanins can appear blue only under specific pH conditions, metal-ion coordination, and co-pigment stacking interactions. Even then, stability is limited. Transition metal chemistry demonstrates that blue pigmentation is chemically achievable when different electronic orbitals are available. Transition metal d orbitals can produce electronic transitions directly within the visible spectrum. Inorganic crystal lattices stabilize these transitions and prevent photochemical degradation. This is why minerals and synthetic pigments often display stable blue coloration. However, biological systems rarely use heavy metal pigment strategies. Metal toxicity, solubility challenges, metabolic cost, and environmental variability make metal pigment strategies risky for living organisms. Evolution, therefore, favors alternative methods. Structural color represents the most common biological solution. Instead of tuning molecular orbital energies organisms tune spatial geometry. Nanoscale periodic structures manipulate light through interference, diffraction, and scattering. Structural color allows blue wavelengths to be reflected without absorption. This avoids excited state photochemistry entirely. Structural blue is especially common because required periodic spacing aligns well with biologically achievable nanoscale assembly. Red structural coloration requires larger periodic spacing, which is mechanically more difficult to produce reliably. Photonic crystal structures represent the most advanced biological optical materials. These structures create photonic band gaps that prevent specific wavelengths from propagating. Blue wavelengths can be strongly reflected simply because they are forbidden from traveling through the material. In atmospheric systems, blue emerges through scattering rather than absorption or reflection. Rayleigh scattering redistributes shorter wavelengths more efficiently than longer wavelengths. The sky appears blue not because air molecules are blue, but because they scatter blue light preferentially. In liquid systems such as oceans, blue emerges through weak but cumulative molecular absorption. Water molecules absorb red wavelengths slightly more strongly than blue. Over long distances, this selective absorption shifts transmitted and scattered light toward blue. In each case, blue emerges not from stable chemical blue molecules, but from selective removal or redirection of other wavelengths. Blue is what remains when red and green are filtered away, scattered away, or prevented from propagating. From a statistical physics perspective, blue often emerges from large-scale averaging behavior. In the sky, trillions of molecules collectively scatter blue wavelengths. In the ocean, trillions of molecules collectively absorb red wavelengths. No single molecule is responsible for blue appearance. From an evolutionary biology perspective, blue represents a compromise between visual signaling and chemical stability. Structural blue allows organisms to display vivid color without risking photochemical degradation. This is why structural blue appears repeatedly across unrelated evolutionary lineages. From a neuroscience perspective, blue is not measured directly. Human vision uses three cone types with overlapping wavelength sensitivities. Blue perception emerges from relative activation patterns rather than absolute measurement. Different species perceive blue differently. Some species detect ultraviolet, others cannot distinguish blue from green effectively. This means blue exists not only as a physical interaction between light and matter, but also as a neural interpretation. Blue is partly constructed by the brain based on incoming spectral information. Philosophically, this leads to an important conclusion. Blue is an emergent property. Emergent properties arise when complex interactions produce behavior not present in individual components. No single molecule is blue. No single photon is blue. Blue emerges when photons interact with matter and are interpreted by a visual system. Blue therefore exists simultaneously as a physical phenomenon and a perceptual phenomenon. It exists as wavelength selective scattering, selective absorption, interference patterns, and neural interpretation. If we step back, we can see blue as a boundary zone. It sits at the high-energy edge of visible light, where organic chemistry begins to struggle with stability. It sits at the wavelength range where nanoscale structural manipulation is most efficient. It sits at the optical range where atmospheric scattering is strongest. It sits at the sensory range, where human vision is most sensitive to contrast against green and red. Blue appears everywhere, not because it is easy for chemistry to produce, but because physics provides many indirect pathways to produce it. The sky is blue because molecules scatter it. The ocean is blue because water removes red. Feathers are blue because nanostructures reflect it. Minerals are blue because transition metals allow it. Flowers are sometimes blue only when chemistry is perfectly balanced. The recurring pattern is clear. Blue is rarely stored. Blue is generated dynamically through interaction. From a freshman chemistry perspective, this topic teaches a larger lesson. Chemistry does not operate in isolation. Molecular behavior is constrained by physics. Biological evolution is constrained by chemistry. Perception is constrained by neurobiology. Blue exists at the intersection of all three. In closing, blue is not an illusion, but it is not a simple property either. It is a demonstration of how the universe produces complex sensory experiences from simple physical rules. Blue exists because photons carry energy. Molecules have quantized orbitals, materials have refractive indices, and brains interpret signals. Blue exists because the universe allows certain wavelengths to survive long enough to reach your eyes. And in that sense, blue is less a thing and more a moment. Summary. Across this entire lesson, we explored one central scientific question. Why does blue appear so common in the natural world when true blue chemistry is actually rare? At first glance, blue seems everywhere. The sky is blue, the ocean is blue, many birds, insects, and minerals appear blue. But when chemists examine natural molecules at the atomic and electronic level, stable blue pigments are far less common than red, yellow, green, or brown pigments. Understanding why requires combining chemistry, physics, biology, and even neuroscience. We started with the most fundamental idea in chemistry and physics. Color is not stored inside objects. Color is created when light interacts with matter and is interpreted by the brain. Light is electromagnetic radiation. When light hits a molecule, electrons can absorb photons and jump between energy levels. The difference between those energy levels determines which wavelengths of light are absorbed and which are reflected. Most natural pigments are organic molecules built from carbon-based frameworks with conjugated electron systems. These conjugated systems allow electrons to move across the molecule, lowering the energy needed for electronic transitions. As conjugation increases, molecules tend to absorb lower energy light, meaning longer wavelengths such as red or orange. This naturally pushes organic pigments toward absorbing blue light instead of reflecting it. That is why many organic pigments appear warm-colored. They remove blue wavelengths and reflect everything else. Blue light sits near the high-energy end of the visible spectrum. High-energy photons are more likely to push electrons into unstable excited states. When molecules absorb blue light repeatedly, they are more likely to degrade through photochemical reactions. These reactions can break bonds, produce reactive oxygen species, or permanently alter molecular structure. Over time, this damages pigments. From an evolutionary standpoint, this creates pressure against relying on blue-absorbing molecules for visible coloration. We then explored anthocyanins, one of the few natural pigment families that can appear blue under specific conditions. Anthocyanins can shift color depending on pH, metal-ion coordination, and molecular stacking interactions. Even then, blue coloration is often unstable and requires tightly controlled cellular environments. This showed us that organic chemistry can approach blue coloration, but only through delicate balancing of electronic structure and environment. Next, we moved into transition metal chemistry. Transition metals contain d orbitals that allow electronic transitions directly within visible wavelengths. This makes blue pigments chemically easier to produce in inorganic systems. Many minerals and synthetic pigments use transition metal complexes to create stable blue color. However, biological systems rarely use this approach because high concentrations of metals can be toxic, difficult to control, and metabolically expensive. Because pigment chemistry struggles to produce stable blue, biology often uses a completely different strategy. Instead of using molecules to absorb and reflect specific wavelengths, organisms build nanoscale structures that manipulate light physically. Structural color works through interference, diffraction, and scattering. Light waves interact with repeating nanoscale patterns, and certain wavelengths are reinforced while others cancel out. Structural blue is especially common because the spacing needed to reflect blue wavelengths falls within the size range that biological systems can easily produce. Proteins like keratin and structural materials like chitin naturally self-assemble into nanostructures that interact with blue light efficiently. This allows organisms to produce bright blue coloration without exposing molecules to damaging high-energy photon absorption. We expanded this idea further into photonic crystals. These are materials with repeating refractive index patterns in 3 dimensions. Photonic crystals can create photonic band gaps where certain wavelengths cannot pass through the material and must be reflected. Some biological tissues naturally form photonic crystal-like structures through self-assembly processes. These structures can produce extremely pure and stable blue coloration using physics rather than chemistry. After exploring solids, we moved into gases. The blue sky is one of the clearest examples of blue appearing without pigments or structure. Atmospheric gases scatter shorter wavelengths of light more efficiently than longer wavelengths. This is called Rayleigh scattering. Blue light is scattered strongly in all directions, which is why the sky appears blue when we look away from the sun. No molecule in the atmosphere is blue. Blue is created through collective scattering behavior across enormous numbers of molecules. Then we examined water. Pure water appears colorless in small amounts, but blue in large volumes. This happens because water molecules absorb red light slightly more strongly than blue light due to vibrational overtone absorption. Over long distances, even weak absorption removes enough red light to shift remaining light toward blue. Scattering then sends that blue light back toward observers. Once again, blue appears not because water contains blue pigment, but because other wavelengths are removed more efficiently. When we combine all these fields, a clear pattern appears. Blue is often not produced by stable blue molecules. Instead, blue emerges when physics removes or redirects other wavelengths. Blue appears when red and green are filtered away, scattered away, or prevented from propagating through materials. From a quantum chemistry perspective, blue sits at a difficult energy range for organic molecules. From a thermodynamic perspective, blue-absorbing molecules are more prone to photochemical damage. From a biological perspective, structural color is often cheaper and safer to maintain than fragile blue pigments. From a physics perspective, blue wavelengths are easy to scatter and easy to reflect with nanoscale structures. From a neuroscience perspective, blue is a perceptual interpretation created by the brain comparing signals across cone cells. All of these systems reinforce the same idea. Blue is often an emergent property. It is created by interactions between light, matter, and perception, rather than stored directly inside matter. This does not mean blue is fake or imaginary. It means blue is dynamic. It exists because physical laws allow certain wavelengths to survive interactions with matter and reach our eyes. Blue exists as an event rather than a permanent substance. Studying blue teaches us something bigger than color. It shows how chemistry, physics, and biology overlap. It shows how evolution often solves chemical problems using physical solutions. It shows how perception shapes what we think reality looks like. And it reminds us that even something as familiar as color can reveal deep scientific principles when examined closely. Thank you for spending this time exploring science at this level of depth. I genuinely appreciate you being here and engaging with complex topics. If you enjoyed this lesson, please like and subscribe. That helps the channel grow and helps more people discover long-form science content designed for learning and relaxation. If you have ideas for future topics, please put them in the comments. Topics about chemistry, physics, space, geology, biology, deep time, or anything else you are curious about are always welcome. And as always, thank you for being part of the Midnight Lesson.